Okay, MaryBeth, I'll let you hit the record button there. It says you got to do it.
Yeah, I've got you going.
Okay, sounds good. All right. All right. Well, good morning, everybody. Let me get the introduction screen up here and present that. All right, there we go. All right, so I'm Robert Bauer, Barren County High School. MaryBeth Cook will do the second session. She's got gene expression and regulation, and I'll do cellular energetics. So basically photosynthesis and cellular respiration. As you know, AdvKY is a trademark registered by the College Board, which is not affiliated with or does not endorse our sessions. If you have any questions, please direct those to Mr. Acampora. Lou Acampora is the Senior Director of Science for Advanced Kentucky. His email is on the screen, and the Twitter and Facebook contact for Advanced Kentucky is there as well. They are our hosts for today. As you are used to by now, through over a year of this, microphones off, videos off. We'll use the chat feature quite a bit, as I think both of us are going to focus a lot more on essays today and maybe some chat involved typing to answer questions. So please participate, and you're only going to get something out of this if you really pay attention and do that participation and kind of self-assess your answers to those essays. All right, there'll be a recording there at that link. If your classmates were not able to join us today, you can share that with them. And let's go ahead and get started. So we're right at 11:32. So we'll go for about 55 minutes, take a break, and then MaryBeth will pick up for us, okay? So photosynthesis and cellular respiration is going to be our focus today. And we'll start with a little bit of general energy knowledge. So I'll show you what I've got over here on the right side of your screen. These are what I call boxes. My kids know them as boxes, but I take those boxes from the content. So the questions are directly written from those boxes, the content. And so if you know those boxes and you can relate that, you can apply it, you got a pretty good chance of getting essays right, getting multiple choice questions right, which is what we know the two components of our exam is going to be. So when it comes to energy, we know that life requires energy. We have to input energy. We take in energy as our food. We use energy through our daily lives. So there has to be more in than out for us to stay alive. So what we are is we're an open system. So we do not violate the second law of thermodynamics. Our energy input exceeds what is lost to maintain all of our cellular processes. So we are an open system. We constantly take in energy. All right. And we use it in this analogy to spin that turbine there. And that's how the energy goes out of our system. If we were a closed system where nothing came in, but we still used up that energy, that would obviously lead down here to equilibrium, which is not good for biological terms as far as energy is concerned. So we need to constantly put in energy to maintain our ability to power our cells. Wrong button. Okay. And we know what we're doing is ultimately that energy that we take in through our bodies is making ATP. Cellular processes that release energy are coupled. So we couple different reactions. So that would be input and output reactions. Those input and output reactions combine to make up metabolism. So the two components of metabolism would be anabolic and catabolic reactions. So to get us started, to just see who's paying attention and thinking real quick on us, I'm going to explain down here an exergonic and an endergonic reaction, which are pretty straightforward. And you've seen those in chemistry if you've taken that class already. And let's do endergonic first. Endergonic is an input of energy. That's where the reactants have less than the product. So we take in energy. Okay, we take in energy. We store that energy. It's coupled a lot of times with an exergonic reaction. That's where the reactants have more, so the products have less. This would be an output of energy. So this is going to happen spontaneously. The free energy change negative, where the free energy change here is positive. It can't happen spontaneously because we can't just spontaneously go up a hill, if you will. Okay, so we know the two terms anabolic and catabolic are what make up metabolism. So the combination of all those reactions in our body make up our metabolic function. So let's break these up in parts. Which one would you say matches catabolic? Would a catabolic reaction, and I haven't defined that yet, I know, would a catabolic reaction match more up with exergonic or match more up with an endergonic reaction? And I'll let you type that in the chat here that I'll monitor to see if we're on the right page. So just type in, does an exergonic reaction match more with catabolic or anabolic reactions? It doesn't have to be a private message to me. It can just be a general message that everybody can see there. So an exergonic reaction over here on the left side, does it match with catabolic or anabolic is the question. So you've got to think back to, do you remember what catabolic and anabolic is? Okay, maybe we need to go back and review what those are. And we'll do that here after everybody kind of has a chance to answer. Okay, so exergonic is releasing energy. So since catabolic also is going to be an output of energy, exergonic would match up with a catabolic reaction. So if you get this one, you're obviously going to get the other one. Endergonic is taking in energy, so that is an anabolic reaction. All right, so endergonic stores, exergonic releases, catabolic is going to break down, anabolic is going to build. So anabolic stores, catabolic releases. Okay, so you kind of get this from here. ATP is made from that ADP to ATP reaction. ATP is broken down as energy is released, energy works. So that's going to be our I guess our cycle is what I should say, of how things work. So let's go up here and look at the structure of ATP. Here's another question for you real quick. ATP has three parts. It has three phosphates. That's why it's called triphosphate. And it has an adenosine base. So adenosine triphosphate. So the three parts are a phosphate, a nitrogenous base, which is right here, and then a 5-carbon sugar. Based on those three components, what kind of molecule, what macromolecule is ATP belong to? So type that one in the chat there. What macromolecule group is ATP, or does ATP belong to? Okay, a couple answers there. Thank you for that. So you're thinking there, maybe nucleotide, maybe nucleic acid. So my question was, what macromolecule group does it belong to? It is a nucleic acid. It is a nucleic acid, which monomer is a nucleotide. So this is a nucleotide, which will make up nucleic acid. So the correct answer is nucleic acid. All nucleotides have three parts, a phosphate, a nitrogenous base, and then a 5-carbon sugar, which the ATP does have. Okay. So moving along here. All right. So this is going to be our basis for our conversation today. So again, these are coupled reactions. These allow for a sequential controlled transfer of energy, as the box says there. And a product of one is generally the reactant of another. So let's look at photosynthesis. If I was going to talk about photosynthesis takes in CO2, takes in H2O, and makes us a carbohydrate while producing oxygen as a byproduct. All right, so these products are the reactants of cellular respiration. That glucose and that oxygen is converted to CO2 and H2O as we make ATP. So again, these products can become these reactants of photosynthesis. So it's just this constant cycle over and over again. And what we'll focus on here in just a second is what goes into what. I'll go ahead and give you a preview of that. A common misconception is Because we breathe in we breathe in oxygen and we produce CO2 in cellular respiration because we take in oxygen and release CO2. That's what comes out of our airways. Those two convert to each other. Well, that's not exactly correct. The carbohydrate actually matches up and is broken down into CO2 as we fix carbon, and oxygen gets that H added to it. And so the oxygen becomes water. So that's things that we'll focus on as we move on down the line here. Okay, so let's go back. All right, this is an overview of the two coupled processes together. So the mitochondria produces that carbon dioxide and water. The chloroplast takes it in, produces oxygen and a glucose, and a byproduct here is ATP. Notice the driving force that makes these reactants into these products is light. That's a driving force. That's not taking light energy and making it into an organic molecule. That's using light energy to drive the process. That's one misconception that we really didn't get on the mock exam. Okay, it's not light energy to organic molecule. It's light energy driving the process or driving the conversion of that carbon dioxide into the carbohydrate. Okay. So now let's move forward and let's see if we can do this question. Okay, this is just an identify. So 2 identifies should be a quick, just what it is. There's no elaboration, there's no explanation, there's nothing here that you have to go into great detail about. Just the 2 words will give you the answer. So let's practice this real quick. All right, here's a cycle. Cycle 1 looks like it's CO2 to an organic molecule. Cycle 2 is organic molecule to CO2. So my question in here is I want you to type me, just say number 1 is this, number 2 is this. So identify the key metabolic process for step 1 and the key metabolic process for step 2. So in order to get that identify point, you have to tell me which one they are, and you have to get both of them correct. So the hint is, we just looked at it in the previous screen. Okay, so I would like for everybody to please do that for me. So what 1 is and what 2 is. Okay, 1's going to be one thing, 2 is going to be another. You have to identify them both, please. Go. One takes CO2 and makes it into an organic molecule, like a glucose, and organic molecules are broken down to CO2 in step 2. I'll give you about 30 seconds more here to type that in. Okay, it looks like everybody's doing a great job on this one. CO2 to an organic molecule, that's carbon fixation, right? So carbon fixation happens in photosynthesis, and then that organic molecule is broken down and release CO2 in cellular respiration, and that's how we get ATP. That's right. So this seems like an easy question on the surface. We'll see if it is. Identify an organism that carries out both processes. Okay, what would be an organism? So just an identify, so it's a one word, or most organisms would be one word, I would assume. So throw me in one. I'll give you 15 seconds. Give me an organism that does both real quick in the chat, please. So plants would be the most common answer, right? And why is that? Plants have to have the organelles that do both. So if they have to have the organelles that do both, that means plants have to have a chloroplast and they have to have a mitochondria. That's a misconception I hope we're long past, that plants only have a chloroplast and animals only have the mitochondria. That's not correct. Animals only have a mitochondria, but plants have a chloroplast to make the glucose, and they have to have the mitochondria to use it for their own energy, because they use ATP to grow and reproduce and produce whatever they're producing, whether that's a flower, vegetable, fruit, any of those options maybe. Okay. All right, onward then. That was just a sample. We don't have to read that. But they do, the second part of that we covered up was an explanation. So when we get a word like explain, we need to go into detail. We need to go into detail about why we identified one as photosynthesis and why we identified two as cellular respiration. Why, or how, in other words, is CO2 getting converted to an organic molecule in photosynthesis, and likewise organic molecule to CO2 and cellular respiration. Okay. So don't miss a point. There's two ways you can miss a point here. One is not answering what step one and step two is, so leaving part out, and then not explaining it. Okay, so we need to get those easy points and get easy points by answering all parts of the question. That's the first thing I'll tell you today. Okay, so right away, let's look at another one. I know we haven't gotten into the meat of photosynthesis yet, but let's see. This is kind of testing to see what we know. So we're what we need to review, I guess, at the same time. Okay, I'll let you read the question as I read it here. To explain the increase in mass of the light-grown plants, identify one inorganic source of new plant mass. Okay, so that's the first part. That's the first part of it. Okay, and then connect it to the cellular process that gives us that increase. So which plant is going to increase mass? Which plant's going to increase mass? That's obviously going to be treatment group number 2, right? It's in the light. It goes from 14 to 32, so an 18 increase. So type that one in the chat. What would be a source of inorganic source of new plant mass? So why is that plant gaining mass? In other words, why did it gain 18 grams? Okay, see what we can throw in there. There's two answers that I would accept here. Challenge yourself to see how well we can do here on these essays. That's going to make the difference in you passing or not. So why did the plant grown in light gain 18 grams over 1 week? Think about what process that plant's going through. Okay. If we think about the reactions, what is that plant taking in? It's obviously going to take water in through its roots. It's going to take CO2 in through its stomata, right? And it comes in through the air. So it has to let that in through the leaves. So either one of those would be an acceptable answer, water or CO2. And then the connection would be, those are going to be incorporated into a carbohydrate by the plant. And because the plant doesn't like just hand out carbohydrates to all of its neighbors, it's going to keep that carbohydrate within its tissue until it's either eaten or broken down by another organism. So that's why it keeps that 18 grams of mass there. Okay. Likewise, what would cause a decrease? So we put a plant in the dark. The other treatment group is we put it in the dark and we end up losing 3 grams. So why did that happen? Is there a reason for that? So same thing, identify the chemical reaction that occurs in the plants to cause it to lose 3 grams of mass. I'll give you about 15 seconds to think on this one. It should almost be the opposite. If one causes it to gain, the other one will cause it to lose, right? Transpiration, Julia, is not a bad guess, but the chemical reaction doesn't occur for transpiration to occur. Okay, that's why that might not be the right road to go down. Okay, but I appreciate you throwing that one in there. Okay. And it's been a long time since we talked about transpiration, so I'm glad you remembered that. The process here that we need to connect it to is, if it's in the dark, it's going through cellular respiration. So it is using up that energy, and it's going to end up releasing carbon dioxide as a byproduct. So it's going to release that through its leaf tissue. So that's how it's going to lose 3 grams. Okay. So some good questions there for us to think about. So let's go ahead and get into this. Okay. Let's go ahead and get into this, and then we're going to talk for a minute, and it's going to be a few seconds before we have some questions to answer. Okay. So photosynthesis, I told you, it's an anabolic process. So that means it builds and it stores energy, but endergonic. Those are the 2 words we were introduced to earlier, right? So anabolic builds, endergonic builds, takes in energy, stores energy. So what is turned into what? So remember redox reactions. Remember redox reactions? I'll try to type these on the screen if I can find my type button real quick. Okay, remember redox. I'm sorry, it's blue. You can see it a little bit. Redox reactions, the mnemonic is Leo says GER, and that's all I'll type. I'll let you listen and write down the rest if you need to. In redox reactions, when I lose electrons, I'm oxidized. And when I gain electrons, I'm reduced. So let's go back over here and look at this question. If I can find my pointer real quick. All right, CO2 is losing that hydrogen. So that hydrogen has electrons attached to it. So that's why it is, hold on. I said that completely backwards. I'm sorry. I'm still trying to find my cursor. That's why I'm getting a little off track there. Anyway, CO2 gains the hydrogen. So CO2 gains the hydrogen. That's why it's reduced. So that hydrogen is stuck in the middle. So technically what we do is we fix that CO2 into a carbohydrate. What part of that, where does that happen? That's what we'll look at in the bottom section. So kind of take your eyes down to that bottom graph and see where CO2 is incorporated into a glucose, or in this case, it's sucrose. And then the other part of that is water loses that H to become oxygen. So when you lose the H, you are oxidized. So water is converted into oxygen. CO2 is converted into a carbohydrate. You've got to know that. You have to understand the process of what's going to what. So we broke it down into 2 processes, the light reactions, and then don't say the dark reactions because it doesn't happen in the dark, but it's the light independent reaction, otherwise known as the Calvin cycle. So in the light reactions, we have these things called photosystems. Photosystems 2 and photosystems 1 take in the light, they absorb the light, and what they're actually doing is making ATP and NADPH. Okay, so a lot of people thought, ATP and NADPH are what the goal of photosynthesis would be. That's what you said in your mock exam essays. Well, that's not correct. At no point in that equation up there at the top do we have a ATP or NADPH. Those are merely intermediates that are actually going to drive the Calvin cycle. Remember, the Calvin cycle happens independent of light. So there's no light that energizes it. Over on the light side, that's the photo part of photosynthesis, because that light is coming in, hitting the photosystems. Those photosystems are using that light to drive the electron transport chains, which makes ATP and NADPH. Okay. Another thing you did not do well on, and we're going to hit on multiple times today, is where these actually occur. All right. The electron transport chain and the photosystems happen in those little stacks called thylakoids. Those thylakoids have membranes where those reactions occur. The Calvin cycle happens in the stroma. So if you remember, I think it was question 5, and we'll look at it towards the end of the day, you were supposed to label where those reactions happen. So that's another thing you need to concentrate and kind of focus on. All right. Hold on. Okay, so we're really going to get going now with part of these. So we kind of did the summary here. So now we're going to subdivide out the light independent and the light dependent reactions. So we'll start with the light dependent reactions. So because they come first. So what happens is this photon hits these little molecules in here, and those molecules are a pigment called chlorophyll. So a pigment is anything that is going to be associated with an interaction to produce a color. So we think when that photon hits it, what's the actual term there? That would be it's going to absorb the light, right? So it's going to absorb all colors except green, and it's going to reflect green, and it reflects green. That's why we see plants as green, right? So the photon hits those chlorophyll molecules, and then ultimately it energizes this electron right here. So it's going to take that electron to a higher energy level. You think of the movement up of that electron as it has more energy, positional energy, and that's basically the start of it. So the light comes in, the photon is the packet of light, and that light's going to be captured by chlorophyll, energizes an electron. So now we have the ability to do work, or we have potential energy, as you see up here. So photon absorbed by the chlorophyll shoots that up to an excited state. What we do with it is now what we're going to focus on after this point. Okay. So if you got questions, throw them in the chat. I'll be happy to answer them. If I'm not hitting something that you don't understand, or if I'm saying something that it completely doesn't make sense, please just stop me and say, hey, can you explain this? That's what we're here for. We're not here for just general stuff. It needs to be personalized if we can make it that way. Okay. So last thing before I go to the next slide, this is the thylakoid. So this is right here, those stacks. You see those stacks? They look like pancakes right here. This is the membrane of that pancake. So this is the thylakoid membrane. That's where that light reaction takes place. This is the photosystem. And then out here in the open, outside of that membrane, that's the stroma. So that would be like the cytoplasm of the chloroplast. Okay. So let's see what that actually looks like. Let's go here. Okay. So we're in the thylakoid space. So this is that pancake. This is the thylakoid membrane here. You see it's a phospholipid bilayer. So membrane. This is the inside space. This is the other membrane. Or the bottom part of that membrane. And then outside of it is the stroma. What we notice is we have a high concentration of hydrogens in here. We have a low concentration of hydrogens out there. Let's type in the chat since it's been a second since we've done that. What does that create? Because we have a high concentration inside the thylakoid space and we have a low concentration in the stroma. What does that create for me? Let's see who can get me just a general vocabulary word. What does it create because we have two different concentrations? Okay, a couple right on it, and that's correct. So that's called a gradient. A concentration gradient is an even better way to say that. So we have a gradient is just a difference, right? It's a fancy word for a difference. So high in, low out. That allows us to do some transport. So you're thinking back to simple diffusion, you're thinking back to active transport, you're thinking all those things. So how does active transport move molecules with the gradient or against the gradient? Does active move with or against the gradient? Does active move with or against the gradient? That's a type. That's a type for me. Or you can say down the gradient, with the gradient, or against the gradient. So remember, it's active transport, so we're using energy, so that's going to be against the gradient. Passive transport would be with the gradient, okay? Or you could say down the gradient, same thing, okay? All right, so our goal, if we go all the way back to this slide right here, our goal of the light-dependent reaction was to take three things. You're supposed to take the light in, and what does the light do? It helps facilitate the reaction that converts water into oxygen, okay? Water into oxygen, and at the same time, it makes ATP and NADPH, Okay, so three things. I need to see how I make oxygen. I need to see how I make ATP. I need to see how I make NADPH, which is a little bit different than NADH. Okay, remember it's got a P in it, so P for photosynthesis. All right, so let's see what those things look like. All right, so the first thing I do is I make oxygen right off the bat. So I bring in the water, and what am I going to do? Let's see if you can get me this word. I'm going to lose that H. Okay, I lose the H, and it splits off from the oxygen. So is that oxidized or reduced? Is that oxidized or reduced? Type that one in for me real quick. You guys have been pretty good about typing for me. Keep that up for these two sessions. That'd be great. Water to oxygen. I lose the electrons off of that water, and technically I lose the H2. So when I lose, remember Leo when I lose, right? I lose electrons, I'm oxidized. So good. So what causes that split? What causes that split? The fancy term is called photo, because the light comes in, photolysis. So photo causes the water to break into the electrons and then the oxygen. So this is the process called photolysis. So photolysis is going to break the water into oxygen. It's going to energize these electrons. Okay, now what do we do with that energy? We have potential energy here. What do we do? Okay, we're going to use it to transport hydrogens across this membrane. Why do we need energy to transport hydrogens across that membrane? Remember, we're going low to high. When we go low to high, that's active transport. So we need this energy to go across the membrane. Okay. So hydrogens across the membrane, and look where these hydrogens go. These hydrogens go all the way down here into this thing called ATP synthase. This is an ACE, so it's an enzyme that synthesizes ATP. It's an enzyme that synthesizes ATP, so that means it takes ADP, adds a phosphate. So as it adds that phosphate, it converts into ATP. How does that? So let's just think of it as a turbine that spins. How does that spin? How does ATP synthase spin? What gives it the energy to spin? It would be these hydrogens. So think about it. We fall down high to low. So that's natural passive flow going from high to low. So that gives that energy to spin. And that energy is used as it falls down through that enzyme. It's going to cause the ADP to be converted into ATP. So we have usable energy here. Okay. We have one source of usable energy that's going to be taken to the Calvin cycle. Okay. So the other source that we have to use is, or make, is NADPH. But the problem is we're out of energy. These guys have fallen all the way down. We're out of energy. So we have to re-energize them, use more light, hits the chlorophyll, re-energizes those electrons. Those electrons move down through here, hit this enzyme called NADP plus reductase. Okay, this is an enzyme that reduces, sorry, hit the wrong button. Enzyme that reduces NADP plus. So when we reduce, we gain electrons, reduce. That makes it NADPH. So now we do NADPH to the Calvin cycle. We do ATP to the Calvin cycle. And I really like this analogy here that we can show. If you look at it, that photon energizes, so up the electron. And as it falls down the first time, it makes ATP. We re-energize it, and as it falls down the second time, we make NADPH. So we use these photons to generate ATP and NADPH, which are going to go to the Calvin cycle to power it, because remember, it happens independently of light. Okay. Questions on the light-dependent reaction that happens in the thylakoid. Questions on the light-dependent reaction that happened in the thylakoid. We got to roll quickly over to cellular respiration, if there's not. Okay, so I'm going to talk about this very quickly. This is the Calvin cycle. This happens in the stroma. The Calvin cycle happens in the stroma. So that's like the cytoplasm of the chloroplast. What does it do? All I'm focused on, the level that you need to know this on is very basic. Okay, what we're going to focus on is let's look at it from this standpoint. Okay. We bring in CO2. We bring in CO2 and we use an enzyme called Ribisco. Okay. We use an enzyme called Ribisco. That's going to mediate the transfer of that carbon dioxide all the way down here into something called G3P. That's basically 3 carbons. I need 6. So what am I going to do? I'm going to do this guy twice. Okay, so I'm going to go 3 carbons once, 3 carbons twice. 3 plus 3 gives me a 6 carbon glucose. Okay, how does this turn? Well, it turns with the help of an enzyme, but the energy to make it turn is ATP, and guess what? NADPH. Okay, ATP and NADPH. So if I didn't have the energy from the light dependent reaction, there's no way I can do the independent reaction. Okay, that's just a quick summary, and I think that's what you need to know from photosynthesis. Okay, questions, and I'll give you just a few seconds to kind of regather yourself after photosynthesis, and I'm going to ask you a question real quick here on this essay. Okay, so I'm not really concerned about all of this. This is a great essay if we have time to do it, but for time purposes, what I want to focus on is this null hypothesis. Okay, we know a null hypothesis means that there's no difference between all the groups. Okay, and you saw that on your mock exam, and they're going to see that. You're going to see that on your actual exam. You're going to have to tell me what the null hypothesis in an experiment is. Okay. Well, it wouldn't be a point if you could just say there's no difference. Okay. You have to say there's no difference in relation to the actual question. Okay. So let's set this up real quick. Read along with me. I'm going to read quickly. So we have a controlled experiment to analyze the effect of darkness and boiling on the photosynthetic rate of incubated chloroplast suspensions. So I have one normal unboiled, I have a dark unboiled, and I have a light boiled. Okay. Just by looking at that, which one of those would be my control? Which sample, 1, 2, or 3, so just type me a number, would be my control in this experiment? Should be able to hit me a number pretty quick. Okay. One's definitely the right answer. Okay. Why is it the right answer? Because it's in normal circumstances, the chloroplast needs light and it doesn't need boiling to interfere with any of its proteins. Okay, dark here, the chloroplast cannot absorb those photons. Boiled here, we're going to denature those enzymes which are needed for the electron transport chain, the enzymes which are needed to convert NADPH and ATP over to the light independent reaction. So we know sample 2 and 3 are the experimental, right? Okay, so we have a dye reduction technique. All right, so instead of using NADPH, we're using something called DPIP. So when DPIP accepts an electron, just like this guy is going to accept electrons, okay? This guy accepts electrons, right? So if this guy accepts electrons, DPIP is also going to be able to accept electrons. It's an electron acceptor that changes color from blue to clear. So basically, in summary, quick, it's going to be blue to clear if it works like it should, okay? So give me your best guess at what a null hypothesis should be for this experiment. Okay. I'm going to give you 45 seconds to write me a sentence. Okay. And then we're going to head over to cellular respiration. Okay, finish up and hit enter for me so we can assess these and get going. So the DPIP is going to accept electrons in normal situations. What would be a null hypothesis? Ten seconds. Okay, I think we're dancing around it a little bit. So I mean, we're getting close. Okay, so remember, null hypothesis would be there would be no. So it should say something like this: there's going to be no difference. Okay, there should be no difference in the percent transmittance in the 3 samples. Okay, so that starts us saying that it's talking about the experiment specifically. So there's no difference between the percent transmittance in the 3 environments, possibly. Okay. So I'm looking and seeing what they're measuring. So percent transmittance would be definitely the dependent variable. And so the only one that works, because there is a difference in percent transmittance here, is I'm saying it's getting blue to clear so I can transmit more light. I know it's getting more clear because more light's going through. Here it's not working because it's in the dark, so it can't absorb light. Here it's not working because those are denatured. Okay. All right. Let's skip over this one for time's sake. Let's go over to cellular respiration. So cellular respiration should be a reaction that is the opposite. So the reactants, remember, and the products would be switched here. So all the way back here. So the products of photosynthesis now become the reactants of cellular respiration. Okay, and the products of this will be the reactants of photosynthesis. Okay, so what do we have? So carbon was converted or fixed in, carbon dioxide was fixed into a glucose in photosynthesis. So we use that glucose and the oxygen, and we're going to use it to make ATP. Well, you don't see ATP on here, but we're oxidizing. Why are we oxidizing? Losing this hydrogen. We're losing that hydrogen and makes it into CO2. So technically what we eat, the glucose, is actually what we breathe out in CO2. It's not what we breathe in, it's what we breathe out. That's actually not a true statement, okay? Because what we eat is converted to CO2, and that's what we have to get out of our lungs, okay? The oxygen is actually converted to water, all right? And that's reduced because it's gaining that hydrogen to become water, all right? So in summary, these boxes are really good. These boxes are really good. It takes you a step-by-step kind of what happens. So instead of reading those boxes to you, we'll just kind of explain what they are over the course of this next few slides. We've got about 15 minutes left, so it goes quick, okay? So first step, glycolysis. Notice where glycolysis happens. Glycolysis happens outside of this mitochondria. Glycolysis happens in the cytoplasm, the cytosol. All right, it converts glucose to pyruvate. That pyruvate, if we have oxygen, goes into the mitochondria. It's broken down into acetyl-CoA, which can go into the citric acid cycle. And then those electrons that are made through NADH and FADH are taken to oxidative phosphorylation. And that's where we get our biggest bang for our buck for ATP. Okay, so you're thinking, what are NADs and FADs? NADs and FADs are the reduced form of basically electron shuttles. So these are energy carriers that we're going to use to move energy from one place to another. And eventually we're going to yield that energy out, or we're going to get the energy out of them to make ATP. Okay. So a lot of steps. What do you need to know? Like, do you need to memorize all this stuff? Clearly not. What do you need to know? I think it's important that you know how the ins and outs work. Like, what do we put in? What do we get out? What do we put in? What do we get out? Okay. So what do we put in? We're going to put in the glucose in glycolysis. We're going to put in the glucose. And we're going to start this process by putting in a little energy. So that's the energy investment. We have to invest a few ATPs to hopefully get a lot of ATPs. So we're going to put in 2 ATPs. What do we get out? We get a pyruvate. So pyruvate is just a 3-carbon. Glucose was 6. So what does glycolysis actually mean? Well, we're lysing or breaking that glucose in half. And what do we get when we do that? We get some NADs and we get 4 ATP. So total, we yielded 2 ATP through something called substrate level phosphorylation you should have heard of before. So you need to know what's made through substrate level phosphorylation. So substrate level, that would imply that it's an enzyme, right? Helping us do that. These NADs are potential energy, but the most potential energy is still in these pyruvates. So where do we go from there? Aerobically, so if we have oxygen, we go into the mitochondria. So oxygen is the key that unlocks that mitochondria. We go through that little transport protein and we go into the mitochondria, okay? If we don't have it, we go through fermentation. So there's 2 types of fermentation, ethanol or lactic acid, all right? Animals go through lactic acid fermentation. So the big question is, why do we do that? Another question we saw in the mock exam. All right, why do we go through this process called fermentation? It's not to produce lactate necessarily, but it's the main reason is because we have these NADs, we need to do something with them, okay? We want to be set up to where we have these NAD pluses the next time we actually have oxygen so we can send them into the mitochondria and get more energy. So what you're doing in fermentation is you're actually recycling the NADs back to NAD plus. So the next time that we get oxygen or next time we have to go through glycolysis, we have some of the inputs or some of the reactants to actually make it happen. So that is the biggest reason why fermentation takes place. It just so happens that lactic acid is a byproduct of lactic acid fermentation, and then alcohol or ethanol is a byproduct of that fermentation, okay? So the biggest part of this is, and I skipped ahead just a little bit pretty quickly. So let's go back just a second. So let's focus on the aerobic part. So aerobically, we're going into the mitochondria. So I'm taking those 2 pyruvates, I'm taking them into the mitochondria. I'm going to go through a conversion step that gets me acetyl-CoA. In that conversion, I produce a little bit of CO2 and I produce another NAD, okay? Those guys go down here to the citric acid cycle. You're thinking, well, what's important here? Well, the biggest thing is I'm getting more NADs and more FADs. So I'm collecting these along the way. I'm basically collecting these in the pathway. I'm getting a bunch of NADs and FADs, which are energy carriers, energy shuttles. In other words, they're potential energy. They're not ATP, so I can't use them like I can use this ATP here, or I could use this ATP made in glycolysis. So I can't directly use those that were made through substrate level phosphorylation. So I've got to take them somewhere. Where do I take them? I take them to the electron transport chain. To go through aerobic respiration than anaerobic respiration and have to go through fermentation. That's why when your muscles start burning, you're going through that lactic acid fermentation. You have to slow down so you can get some oxygen. So we go back over here to this oxidative phosphorylation or aerobic respiration. So what happens? We've got NADs and FADs. We can't use them, so we're going to try to use them here. All right, so this NAD, NADH, is going to an NAD plus. We've learned already when we lose electrons, that's called oxidation. This FAD is picked up a little bit later down the chain. So it goes from FADH to FAD. Both of those are oxidized. This is oxidized at 1. This is oxidized in between 1 and 3. So at 2, we'll say. Okay. So that's why the electron transport chain, I've got a big red box here saying oxidative. So the oxidative part is popping those electrons off, and look again, we have a gradient. So we're pumping them from a low to high concentration, and we're establishing a gradient. So we pump 1 here, we pump 1 here, we pump 1 here. So every NAD pumps 1, 2, 3. Every FAD pumps 1, 2. So we're pumping those hydrogens across. Why are we pumping them across? So they can come down here and fall through, as we've seen here, ATP synthase, they can fall through. So we're going from a high concentration to a low concentration. And as that hydrogen falls, it spins ATP synthase just like it did before. So that's why it's called phosphorylation. When we add a phosphate, it's called phosphorylation. So this is oxidative phosphorylation as opposed to photosynthesis, which is photophosphorylation because we're using it for the light. And again, ATP is just an intermediate in photosynthesis, but it's the actual product here in cellular respiration because it's what we want to get. We want the ATPs out, and there's where you see the outputs. Okay. So we get 32 to 34 output of ATP made through oxidative phosphorylation, where before we were using enzymes and substrate level phosphorylation. Okay. So we put in some NADs, we put in some FADs, and we get out ATP. Okay. So for every one NAD you put in, you go 1, 2, 3. For every one NAD, you pump across 3 hydrogens, so 3 fall back down. So I get 3 ATPs for every one NAD. I get 1, 2 to pump across, so 2 to fall back down. So I get 2 ATPs per FAD. All right. I know this is quick. I mean, you probably spent 3-4 weeks on it in class, maybe more. And we're doing this in an hour. So it's just a very quick run through. Hopefully we're hitting some high points, things for you to remember, things that you're going to have to go back and study on your own. Okay. So let's finish with a couple questions. I've got time for maybe 1 or 2 questions here, and we're going to turn it over to gene expression. So this is a good kind of connection between MaryBeth's session and mine here. So based on the data, identify a gene that is most likely to encode a protein that is an essential component of glycolysis. Okay. So you don't even have to provide support for me. I'm going to make it easy. However, on the test, you would have to do that to get the actual point. So give me gene E, F, G, H, or I, or J. So which one of those 6 is most likely to encode a gene that is in a component of glycolysis? So give me a good guess, if nothing else. Okay, so I've got some G's in there, a lot of G's. G is right. Guess what? You need glycolysis in every single, it's the first step of cellular respiration in every organism. Every single organism has glycolysis. That's how prokaryotic organisms make ATP, is through glycolysis. They don't have a mitochondria to make more. But your liver, your heart, your brain, your kidneys, your pancreas, your skeletal muscle all have to go through glycolysis before they can even think about going through the rest of cellular respiration and ending with oxidative phosphorylation. Okay, so this is a good tie-in to next session as well. The researcher observed that tissues with a high level of gene H did not always have gene H protein. So I got the messenger RNA, but I did not get the gene. Anybody tell me why, to kind of preview as a maybe a pre-quiz for next session to kind of see what that would be like? Why could we get mRNA but not actually get the protein? Because we know it goes from DNA to messenger RNA to protein, right? Well, why would that be the case? Any good guesses there? A mutation in the transcript would still get me a protein. It might not be protein H. A good guess, Abby. Good guess. Anybody else want to try real quick? Something has to keep it from being translated, right? Something has to be keeping it from translating it. So it could be that it's not translated. So it could be RNA interference. interfering with that transcript, or it could be that it actually is protein is made, but it's degraded down real quick. So I'm sure MaryBeth will talk about those, and I will end with this real quick. Right. So as these seals and penguins dive, all right, as they dive, you can see their blood lactate levels. So it says, based on the data, propose a hypothesis to explain the change in blood lactate levels in the Weddell seals for dives lasting longer than 20 minutes. So I'm right here. At 20 minutes, it looks like their lactate levels go way up. Why do you think that's the case after 20 minutes of a dive? Give me that one, and then we'll call it quits, and I'll hang out here during your break to answer any questions. What's the reason that they are going to have a ginormous jump in blood lactate levels? Let's see who can finish this out today. 15 seconds. So I don't think we can dive for 20 minutes, right? But those seals and penguins obviously have special abilities that allow them to do that. But after 20 minutes, they're going to run out of oxygen, right? So for sure, they're going through anaerobic respiration. So that would send them into fermentation, which means they're going through lactic acid fermentation. The product of lactic acid fermentation would increase their lactate levels in their blood. So fermentation, as Julia said there, would be the correct answer. That's right. I appreciate you guys. Good luck on your exam. You got a quick break there, and MaryBeth will jump on here in just a few minutes, okay? So good luck to you, and I'll turn it over to her.
Okay, guys, we'll go ahead and get started. So this unit, I am going to focus on mainly just on gene expression. So I think Robert did a review session on the central dogma, and he kind of went through the details of those processes. Today, hopefully we'll just kind of review and recap what gene expression is and get you some of those details. So my first question for you guys, if you'll go to chat, what pops in your head when you see the words gene expression? So what are we talking about if the question has gene expression? Okay, anybody else want to pop in before we kind of talk about what gene expression is? So think about gene. So gene is a section of DNA that we're going to make a protein from. And so that expression is going to be the production of a protein. And so going from DNA to a protein does include mRNA. So if somebody said mRNA. And then what two processes allow gene expression to occur? So how do we go from DNA to actually making a protein? So again, type into chat what processes occur to go from DNA to protein. All right, good. So transcription, translation. And so why do you guys think it would be important to be able to regulate gene expression or regulate what proteins get made and what proteins don't get made? So what would be important to a cell that it can actually control what genes are being made into a protein and which genes are not? Okay, so I'm just going to move on so we can get through some of this. The importance for cells to have the ability to regulate gene expression, you're going to see, and if you do think back, you guys knew it was transcription and translation. Those steps, it requires a lot of resources and sometimes energy. And so if a cell can regulate when it's actually making a protein or not, then it can save resources and it can save energy for something that it actually does need at that particular time. And so, especially like prokaryotic cells, those that are really good at regulating their gene expression as a result of what's going on in their environment, they're going to have a selective advantage compared to other cells that aren't so good at doing this. All right, so out of the steps that you guys gave me for chat, what step would be the best step to regulate gene expression and really help the cell in conserving energy or conserving resources? So out of the steps that you gave me, which step do you think would be the best place to regulate gene expression? All right, so transcription is our first step. And so that's definitely going to be the best place. So if we're needing to shut down a process, then that very first step would be the key step to do so. All right, so here's just a picture. I think it is important that you guys do remember what transcription is, translation, and just make sure that you're not confusing those terms. So I think that's usually one of the most common mistakes is getting these two words confused. So kind of think about it, DNA for us, especially, we have a huge amount of DNA. And then we can make a transcript or a smaller version, a more condensed version of that information for a particular gene into RNA. And, chemically talking, DNA and RNA are nucleic acids. And so, we're in the same language. But then to go from RNA to a protein, we're changing chemical languages. So we're going from a nucleic acid to a protein. And so that needs translation to do so. So hopefully that'll help you kind of keep the words separate. I've seen them give you a specific gene and they were making a specific protein. And then they asked students to circle which step was transcription or which step was translation. So that's something, knowing those steps and what we're creating at each one, I think is really important to know. The other thing that I think is important is to kind of keep this, how does this stuff happen in a prokaryote and how does it happen in a eukaryote? And so if you guys will go use the chat feature and tell me where transcription happens in a prokaryote. So where does transcription, taking DNA and creating an RNA transcript, where does that happen in a prokaryote? All right, so there's several differences between prokaryotes and eukaryotes, but the key difference is that prokaryotes do not have a nucleus surrounding their DNA. So to go from DNA to RNA in a prokaryote, that is going to happen in the cytoplasm. Um, so DNA to RNA in a eukaryote. So our cells are eukaryotes. So where would you tell me that happens in a eukaryote? Transcription. All right, good. So this is definitely going to happen in the nucleus. Remember, our DNA, we want to keep it safe from hydrolytic enzymes in the cytosol. And so our DNA does stay kind of protected. It's safe in the nucleus. All right. And then I'm just going to kind of go over translation is going to happen in the same place, whether we're talking about a prokaryote or eukaryote. So can you tell me the specific location where translation occurs in any cell type? So translation happens here. So give me a specific structure of where translation happens. I'll give you one more clue. This is where protein synthesis, right? So translation, we're taking the RNA and making a protein. So where does protein synthesis happen in a cell? All right, good. So I would want you to be more specific. So instead of just cytoplasm, ribosome specifically is where translation is going to happen. Good. So before we start getting into kind of details of gene expression regulation, this does give us an opportunity to talk about feedback. And so feedback is such an important concept to understand. It can be applied to so many different topics that we've discussed this year. And so this is just yet another one. And so a lot of times feedback is going to help, negative feedback is going to help in maintaining homeostasis or maintaining a balance. It's going to help a cell kind of just be more efficient, I think, at doing a particular job. And so regulating its genes is going to allow it to save resources and save energy for other things that are maybe more important at that particular moment in time. So the diagram that you're seeing is showing you two ways that tryptophan. So tryptophan is going to be a gene regulation pathway that we're going to talk about with some details. Tryptophan is really important in for prokaryotes. They need that. It's an important amino acid for them to create proteins that they need. But once a bacterial cell has tryptophan, there's no need to continue making it. So once it has it, that tryptophan can feed back and stop an earlier step. And so if you kind of look, there's 3 different enzymes that are facilitating the production of tryptophan. And so once the cell gets it, it can kind of feed back and then quit going through all these steps and using all those resources for something that it already has. So this is a quick way for the cell to respond. If it finds that it, has enough tryptophan, it can quit making it. But then that tryptophan can also feed back and stop these genes from going through transcription and translation to make these enzymes. And that's going to have or provide a longer term response for the cell. And so to get into gene expression, prokaryotes and eukaryotes differ quite a bit. And so prokaryotes have a specific structure that allows them to regulate their gene expression. Do you guys remember what they have? So if you'll go to chat, do you remember the structure that they have that allows them to regulate their gene expression? And it is completely different from what we have. Okay, I'll see if I can give you a hint. So it has three main components. It has a promoter, an operator, and then the genes that it's trying to regulate. So do you know what we call the structure in prokaryotes that contains a promoter, an operator, and the genes it's trying to regulate? Okay, so someone said ligase. Ligase is an important enzyme in DNA replication. So it would help seal up Okazaki fragments. But thank you for trying. So anybody else want to try operator, promoter, and genes that we're going to regulate in a prokaryote? Okay, so what I was looking for is an operon. And I think it is really important if you're seeing a question about an operon, then your mind should automatically be going to a prokaryote and thinking about prokaryotic characteristics. So there is these three components. That's what you're going to see. There's two main operons that I feel like do come up more often on AP questions and that, you know, I cover in my classes, but bacteria have a lot of operons. And so I think this is definitely something they can mention an operon that maybe you weren't exposed to in class. But as long as you know these three components, I think you can think through those questions. So promoter. So obviously this is going to be a region of your DNA, all of these things. So you have promoter, operator, and then the genes. So this is all sections of DNA. Do you guys remember what will bind to a promoter? So the whole point of gene expression, we've got to go from DNA, transcribe it into mRNA, translate it into a protein. So what do we need to bind to a promoter so that transcription can start? So it's not going to be a primer. So primers are usually signals to start DNA replication. So this is an enzyme. There's a specific enzyme that's going to facilitate transcription. Okay, so you're close, Sydney. So we're trying to make RNA transcript. So instead of DNA polymerase, it's going to be RNA polymerase. So RNA polymerase is going to start the promoter. Operator is essentially the operator. So this is where we're going to be able to actually regulate gene expression. And so there's going to be an important protein that can bind here that actually allows it to operate and say, yes, we're going to transcribe these genes, or no, we need to stop transcribing these genes. Anyway, remember what binds to the operator? So if you do, you can type it in chat. Okay, good. So this would be a repressor. So repressor can bind to the operator. If it's bound, obviously transcription is repressed. And if it's not bound, transcription can actually occur. And then, you know, the whole point of this, we have to have those genes that we're wanting to make proteins from. So those are going to be part of the operon as well. So what we're going to look at, we're going to look at the TRP operon, tryptophan operon, and the Lac Operon. And both of these have negative control in case you're seeing that word describing them. And so what exactly does that mean? Both of these operons that we're going to look at can be switched off by the active form of the repressor protein. And so I just want to give you a picture of each. You know, I think this is very common. They may show you a picture and then have you, you know, answer a question about it. So tryptophan, like I mentioned earlier with that feedback slide, is an important amino acid that bacteria need pretty much all the time. And so we would want transcription to be occurring. So we'd want those enzymes created so that tryptophan can be created. And so this operon is typically going to be on all the time. And so what you're seeing, here's the promoter region, here's the O for the operator that I was telling you about, and then here's those genes that we need to transcribe and make into enzymes so that the cell can actually make tryptophan. And so once the cell has tryptophan, then tryptophan is going to act as an allosteric regulator. So that repressor protein is going to be inactive most of the time so that this cell can create tryptophan. But once it has enough tryptophan and these two interact, tryptophan will bind allosterically, changes the shape. So if you kind of look, here's our inactive repressor with no tryptophan bound. And then once tryptophan binds, the repressor changes shape so that now it can go up here, dock to the operator, and now we're going to turn or shut off transcription. So Rna Polymerase is bound at the promoter region, but it can't get past the operator now. And so therefore all these genes do not get transcribed and they don't get translated. And so that's important. That cell, that bacterial cell, has either created enough tryptophan or now its environment is supplying tryptophan. And so there's no point of going through that step of transcription, translation, spending all those resources, spending all of its energy making something it already has. Once you know, that tryptophan's used up from its environment or from within the cell, then this last tryptophan would be used. And once it's used, you know, it's removed from the repressor, it goes back to its inactive form, and we're back to this picture. Polymerase is bound to the promoter, and it can move past the operator because the repressor is not bound, and we can transcribe and translate all these genes that will eventually make enzymes that's going to actually help create tryptophan again. So again, I can't stress it enough. These operons where all the genes are grouped together that are going to create this one product or help in creating this one product only happens in a prokaryote. So operons are only in a prokaryote. All right, so the next picture I have is another example of an operon. Very common is the Lac Operon. And so when we're talking about Lac Operon, we're talking about lactose. So lactose is a disaccharide, and so it can be an energy source for bacteria, but it's not the preferred energy source. So bacteria prefer to have glucose, as most cells do. And so if glucose is in their environment, they're going to use glucose to get their ATP. And if lactose is present, great, they can break it down. But again, glucose is going to be their ultimate source. So what's going to happen is the genes that need to be transcribed to make enzymes that are going to help break the disaccharide down, those are typically going to be off unless lactose becomes available in the environment. So this is going to be an operon that's typically off, but can be inducible operon, or turned on. And so the active form of the repressor will be attached to the operator most of the time. And so RNA polymerase obviously can't get past it and will not spend time or energy transcribing and translating genes to make enzymes to break down lactose if lactose is not even present in the cell. So if the environment changes, lactose is now available. A version of lactose, allolactose, will bind to that repressor, changes its shape. It's no longer going to bind to the operator, and now RNA polymerase can move past the operator, transcribe and translate those genes. And so now the cell has enzymes that's going to help break down that lactose into monomers, and the cell can actually use that for energy. So two examples, these are your most common ones. But again, if you look at the picture, you have that promoter region where the RNA polymerase binds. You have the operator where the repressor can bind or not. And that's what's really going to help regulate whether these genes are expressed or not. And then you're going to have several genes. So like this Lac Operon only has 3 genes. The Trp Operon had 5 genes. So you've kind of, that's what you would want to look for and kind of pay attention to if they're giving you a completely different example. The other thing that I do want to stress and point out is kind of upstream from these genes that are going to work together to do a job for the cell, you do have a specific gene that's going to make that repressor protein. So that's why that's also included in this image. So you have the gene that's going to make the Lac repressor protein. So I did want to just kind of give you an image comparing the two. For the Lac Operon, which is an inducible operon, the active form of the repressor doesn't have anything attached. So it's kind of active all the time. And then when lactose becomes present, it binds, changes the shape so that it can no longer bind to the operator and transcription can occur. The Trp operon is different. So it's going to be active all the time. So we'd have an inactive repressor. And then once the cell created enough tryptophan that can bind, that forms the active repressor, which will bind to the operator and shut down transcription. All right, so I do want to point out, so the Lac Operon can also follow positive gene regulation. So I was telling you that bacteria prefer glucose as their energy source, but if lactose is present, they're definitely not going to pass up that or pass up using it as an energy source as well. And so what I walked you through with the Lac Operon applies. So, you know, even if glucose is present and then lactose shows up, then we would want this operon to be kind of open and available to transcribe and translate the genes to break down this lactose energy. Here's what kind of makes this different. So say the cell finds itself in a situation where it no longer has glucose, its preferred energy source, and all it has is lactose, which it doesn't prefer, takes more energy to break down the disaccharide compared to glucose, a monosaccharide. And so what's going to happen is that cell, when glucose is scarce, CAMP is going to accumulate. So there's no longer going to be an easy access to ATP. We're left with adenosine monophosphate. And so CAMP will be the signal to dock to another important protein. And so we have a CAP activator protein, and its job is to also bind to the promoter region if CAMP is present. So CAMP is a signal that glucose is scarce, docks to the CAP, and then that unit together will dock to the promoter. And then I kind of think of it as an extra big flag to get RNA polymerase to this promoter so that we can increase the rate of transcription and increase the amount of enzymes that are going to help digest and break down what is currently our only source of energy, the lactose. And so this is an example of positive. We're kind of increasing the rate and the amount of the gene products being produced. And so CAP is one of those, it's kind of generic. This could be used for a lot of different operons if we need to, if the bacteria need to increase the rate or the amount of a gene being made into a protein. All right, so before I move on to any, to eukaryotic regulation, does anyone have questions on prokaryotes and operons? So this is it. Prokaryotes regulate gene expression at the transcription level using operons. That would be the big thing I would want you to hang, hold into your head. And, any of these images I think are fair game for questions. All right, so I have about 27 minutes to go through eukaryotic gene expression, and then I have some multiple choice questions for you guys to think through. So eukaryotes do not have operons, so I can't stress that enough. That is the big difference in gene regulation. We do not have operons. So we have a lot of different chromosomes. So we as humans have 46 compared to that bacteria that I've been talking about has typically one chromosome. And so, a lot of times the genes that are going to either create maybe one enzyme, they could be located on multiple chromosomes. So how do we coordinate and get all those genes to go through transcription and translation to make their polypeptide chains so they can function together as one enzyme? And so our cells can regulate gene expression at multiple levels. And you're kind of seeing that shown in this diagram. I'm going to walk through each component and then we'll practice with some questions. So again, our cells, why is it important that they regulate gene expression? It's going to allow them to respond appropriately to internal and external signals. And then for our cells, regulating gene expression is really important because we're multicellular and we have cells that have different jobs. And so really quick, if you'll go into chat, so we have specialized cells. So think like liver cells versus a heart cell. Do we have those different type of cells because differential gene expression allows them to behave differently as a heart and a liver cell? Or do those two different types of cells have different genes inside of them? So go to chat and just tell me, do we have different or special cell types because of differential gene expression or because they have different genes? And so again, do you have specialized cells? So heart cells, liver cells, skin cells, do you have those because of differential gene expression or because those cells have different genes in them? Okay, and so I think this is one where AP questions are going to try to trick you. And so I want you to think back. Every single one of us started out as one cell. So sperm and egg united, and we all started as a zygote, one single cell. And then that one cell started dividing by mitosis, and then those divided by mitosis, and now we're trillions of cells big. And remember, the key to mitosis is that we're going to produce identical, genetically identical cells. And so the reason that we can have a liver cell and a heart cell and a skin cell with completely different jobs is because they have the same genes, but they don't always express those genes. So this differential gene expression allows your skin cell to create proteins to allow it to function as a skin cell. Your heart cell can just produce the proteins that it needs to function as a heart cell, and so forth. So differential gene expression is how we get specialized cells. Every single one of our cells has the same genetic information. All right, so the first level of where eukaryotes can regulate genes is something called epigenetics. So I think this is a field of biology that's growing. There's still a lot that we don't fully understand about it. But we are back. So you're seeing a picture of a chromosome here. So this blue chromosome. And it coils up. And so remember, this is an important difference between prokaryotes and eukaryotes too. Our DNA wraps around histone protein. So that's what these white circles are. And we can have chemical groups attach, and that's going to either allow kind of some space, some separation of your DNA, or it's going to cause it to condense up really tightly. And so acetyl or acetyl groups are what attach to your histone proteins, and that's going to prevent your DNA from coiling up really tightly. And so if your DNA is nice and loose, then RNA polymerase can get in there, transcribe those genes, and make proteins. And so you may see this referred to as euchromatin. So that EU prefix means true. And then chromatin, remember, is kind of that loose, messy form of DNA during interphase of the cell cycle, or a non-dividing cell would, their DNA would be in chromatin form. And then the opposite of that is if we add methyl groups, so this is called methylation, to the histone proteins, then your DNA is going to coil up really tightly. And so if it's coiled up really tightly, the likelihood of RNA polymerase getting in there and being able to transcribe those genes is pretty unlikely. And so this is called heterochromatin. And a way to remember this, I'm always big on letters. So the H and the C. So heterochromatin, think of highly condensed DNA. And so if it's highly condensed, then transcription can't happen, and we're not going to express those genes in a protein. My Google Slides should be available at the website Robert showed you guys at the beginning of this. And so I did just, there's a link to kind of general, more information about epigenetics, and then just a nice application of how epigenetic treatment might be used for depression. All right, so if our chromosomes, if we're not regulating gene expression at that level, then we can regulate whether transcription happens. So this is in us as a eukaryote. You're going to see, again, a promoter region. You're going to see the gene of interest that we're trying to transcribe. And then way upstream of that section, you're going to have kind of this enhancer region where activators can bind. So if these activators bind, then that's going to be kind of signals to draw in transcription factors. Those bind to the promoter. These mediator proteins are going to come in, and then that's all going to be a big signal for RNA polymerase to come in and bind to that promoter and transcribe those genes. So this is definitely different than what we saw in prokaryotes. Again, it's signaling. These activators are proteins. We have lots of these activators would interact with the mediator proteins, and really they call this a transcription initiation complex. So all these components coming together to ensure that this particular gene gets transcribed due to whatever conditions the cells found itself in. This is also going to be important. Having these activators and having specific transcription factors are going to help this particular gene that you're seeing in this picture. But let's say there's another gene that needs to be made at the same time so that that polypeptide chain can interact with this polypeptide chain. So by having the same transcription factors, that would allow both genes to get transcribed at the same time. So we don't have ours grouped together like the prokaryote did. but by having the same general or same transcription factors and maybe the same activators, we can get multiple genes expressed at the same time. Here's just another picture, but it takes us to really kind of trying to drive home that all your cells have the same genetic info, but they're not going to express all the same genes. And so this picture is showing you two genes. Dopamine is a neurotransmitter. It's a really important chemical messenger between neurons. And then keratin is an important kind of gives structure and rigidity and really allows your skin to act as a barrier. And so the nerve cell is going to have activators. So if you kind of look at this enhancer region, they're color coding it for you to think about these processes. So the nerve cell is going to have specific activators for just the dopamine gene and not have the activators for the keratin gene. And so all of these will bind. That's going to help send those signals for transcription factors to come in, the mediator proteins, and then the nerve cell will only transcribe the dopamine gene. It doesn't have the gene will never be expressed in a skin cell. So again, both cells have all the same information, but it's due to this differential gene expression that a nerve cell functions as a nerve cell and the skin cell functions as a skin cell. All right, so that's how we can kind of regulate. That's what allows our cells to be specific. And then another way that our cells can regulate gene expression is through, once the transcript gets made, there's post-transcription modifications that occur. And this is called RNA processing. And so sometimes an mRNA, pre-mRNA transcript may get created, but if the 5' cap's not added, the poly A tail's not added, and the introns aren't cut out, that mRNA may never go on to be, it may never leave the nucleus to be translated. And this kind of goes back to where Robert ended his session. That could be one answer to why you may have the mRNA produced, but you never get a protein made from it. So if this RNA processing doesn't happen, we're controlling gene expression by never allowing the mRNA to go to the ribosome and be made into a protein. Kind of just another picture to tie in and review the exons and the introns, this alternative splicing. So what sections are an intron, what sections are an exon, and what exons get put together just gives our cells a lot of variation. And so exons 1 through 5 is what we have in this particular picture. If all of those exons get spliced together, we're going to get this protein. But then the next time, maybe exon 3 is not included. And so 1, 2, 4, and 5 is going to create a different protein. And then the last, we could, knock out number 4 and 1, 2, 3, and 5. And that allows a totally different protein to be made from this section of DNA. So how does alternative splicing occur? There's two main ways that you may see or have questions on. So one are just ribozymes. And so this is where RNA itself in that intron section can function as an enzyme and cut itself out. Or you may see pictures with this spliceosome. And so spliceosomes consist of small nuclear RNA molecules. That's what that little red kind of structure is. And a protein, we call those two things together a SNRP. Add that to some more proteins, and we kind of create this larger structure called a spliceosome. So what you see occurring is this single-stranded RNA molecule, small nuclear RNA molecule, part of the SNRP. If it can base pair with bases in our mRNA transcript in that intron, then that's going to be a signal that that is the correct section to cut out. And so it'll cut out the intron, and that allows the exons that we want expressed to be expressed together. So let's say your cell has created mRNA, but the environment's changed. It no longer needs that mRNA to be expressed into a protein, and we can stop the process here. So even though transcription's happened, we can still stop that gene from being expressed. And so this is something called Rna interference. Robert mentioned this for that last question. And so this is where we also have small RNA molecules. They can either be called microRNA molecules or small interfering RNA molecules. For the most part, their job's pretty similar. So they can either, as a single-stranded RNA molecule themselves, if they base pair, so if about 6 bases pair up to our mRNA transcript, now we have a double-stranded RNA molecule that the ribosome can't attach to. And so if ribosome can't attach, translation doesn't happen. So that's one way we can just block translation from occurring, because now we've created this double-stranded RNA molecule. Or these molecules can degrade our RNA, kind of chop it back up into nucleotides, and then use those nucleotides to make an mRNA that it actually does need at that moment in time. So again, we've gone through the step, the cell went through the step of transcription, it processed it, it's left the nucleus, now the environment's changed, but we can still shut down and prevent the protein from being produced. And the last way we can, even our cells can make proteins, but then too, the environment can change. It no longer needs that protein. And to shut it down, we can essentially flag that protein for destruction. And so here's your protein. It's folded. It's doing its job, but the environment changed. We don't want it to do its job anymore. So that protein can be flagged with ubiquitin. That flag sends it to a kind of a protein complex called a proteasome, and that will uncoil our protein and kind of chop it up. Those amino acids can then be used to make a protein your cell does need at this particular moment in time. All right, so the last thing that I just wanted to tie into, so we went through all the ways that our cells as a eukaryote can regulate gene expression. So does the protein get made or not? This is really important in cell differentiation and development. So again, this is what I mentioned earlier, the sperm and egg combine. This is what we all started as a zygote. And inside that cell, there's going to be all kinds of chemical signals or cytoplasmic determinants. So when that cell divides, you can kind of see that the orange circular cytoplasmic determinant ended up in one cell. The green triangular cytoplasmic determinant ended up in another. And those chemical signals are essentially going to help tell that nucleus, hey, we're this type of cell, we're only going to express these genes and we're not going to express these other genes. And so that's what's going to help kind of determine those cells to become a specific cell type. And then as you continue to grow and become more cells, then each cell can send out those signals to nearby cells. And remember, we call that paracrine signaling. Those signals would dock to receptors on the cell's membrane. And then you're seeing the blue arrows to remind you of a signal transduction pathway. And then the response is going to happen in the nucleus where genes get turned on or genes get turned off. And the last thing, we've got about 9 minutes. I just wanted to show you this. I remembered I've seen a question, but I couldn't find the actual question. And so this one's just a great example of kind of that cell differentiation and final determination of a cell. So kind of as we start, you know, as an embryo, we have cells that don't have specific jobs just yet. As those cells continue to divide and develop, this particular cell becomes determined to be a myoblast. And this is going to be a cell in your muscles. And so once it's become determined, this gene for MyoD is now turned on. the gene's transcribed into mRNA and then translated into a protein. That protein, MyoD, is going to act as a transcription factor. And so once it's made, it can now bind to another promoter region for a different gene, transcribe, translate. That makes a different transcription factor, and then that turns on these other genes. So it kind of just helps facilitate and say, yes, we're muscle cells, and then it's only going to create proteins necessary for it to function as a muscle cell. So I just kind of wanted to stress that this, sometimes we can activate one gene, and then that facilitates the activation of other genes for a particular cell type. All right, so let's see what you guys have learned. If you'll go to chat and just throw in the correct letter. So which process will increase transcription? All right, very good. Looks like we're in agreement. So B is the correct one. That one's going to increase the rate. I'm going to skip and I'm going to come back if we have time. So here's another multiple choice. If you'll put A, B, C, or D again. So which of the following statements best explains how protein X regulates gene expression? All right, so it looks like we're in agreement again. So once protein X is produced, it's going and docking to promoters of these different genes. And when it is docked to them, you can see that it is being transcribed. So it's promoting or allowing or activating transcription. All right, let's see what you guys remember about operons. So this is the Lac Operon. I'm not sure if you can see. But in the question, I just kind of want to stress, sometimes they give you a lot of answers or they give you a lot of guidance sometimes to answer a question. So really pay attention to what they're providing you with. So when lactose is absent, the operon is inactive because a repressor protein binds to a specific site in the Lac Operon. When lactose is present, lactose binds to the repressor protein, causing the repressor protein to dissociate from the binding site. In the absence of glucose, which is preferred energy, the protein cap will bind to the regulatory site near the Lac promoter to activate transcription of the Lac Operon. And so they've given us a little different than what I showed you, but the repressor is a circle, lactose is two kind of hexagon structures, cap triangle, RNA polymerase is an oval, and mRNA is a squiggly line. So in the diagram below, the horizontal line is our operon. The bent arrow shows us where transcription is going to start. And so which diagram is showing us where lactose is available and glucose is not available? So A, B, C, or D, tell me in chat which one best shows an operon where lactose is present, glucose is not present? So kind of think about, this is what I talked about. The preferred energy source is gone. So no glucose. The cell has got to utilize the lactose that is present. And so how can it do that the best? Which picture? And again, and maybe you can't see, but they did tell us that lactose will bind to a repressor protein. So if lactose is present, lactose will bind to a repressor protein. So that would be my first kind of look at, because they want us to pick the diagram that's showing where lactose is present, glucose is absent. So where do I see lactose bound to a repressor? So I've kind of got A here and I've got C. So I'm kind of torn between the two. If you remember, this is one of those cases, the preferred energy source is gone. And so this cell now only can utilize lactose. And so it needs to transcribe and translate these genes like crazy so that it can have an energy source. And so CAP needs to be present in this case. And so between A and C, CAP is bound to our operon in C only. And so RNA polymerase is allowed to move down the genes and transcribe it into mRNA. So C is the correct answer. And so Robert saved me. He gave you guys this question. But let's talk about B really fast and see, you guys didn't know what to say at the beginning when he asked this. So tissues with high levels of gene H mRNA, they didn't always have gene H protein. So could you tell me how we could make mRNA, but not necessarily make the protein for it? How can the cell regulate that? So again, use chat. There's about 3 different answers here, I think. So how can a cell go through transcription, but not go through translation? All right, so anybody getting ready to hit send? I don't want to interrupt. So it's not going to be a mutation level here. So we'd still, even with a nonsense, you would make maybe a partial protein. I guess maybe sometimes you do have a start and then a stop, but that's usually not what's happening here. So if you make the mRNA, and it's correct, then remember I told you guys that the mRNA may not go through the processing, which allows it to leave the nucleus. So maybe the mRNA got made, but it never left the nucleus to be translated. If it's not translated, remember mRNA, once it's made, it can either be blocked by the small RNAs or it can be degraded, so kind of chopped up and the nucleotides reused, or maybe it did make it to the ribosome, but that protein didn't get made, or the protein got made and then it just kind of immediately got degraded. So that could be an option as well. And that's why we don't see the protein accumulate in this particular question. All right, guys, I didn't have time to go over slides 22 and 23, but you guys can have access if you go to the link. I also just added some extra videos from Bozeman. I think he does a good job of recapping the information. So these videos are linked there if you feel like you need that extra help. So I'll stick around. If anybody has questions or, wants to ask in the chat, I'm happy to answer those for a little bit. Otherwise, thanks again for joining, giving up a beautiful Saturday, but keep pushing through. I think those that really commit and stick it through for 2 more weeks usually do see the most success on the test day. So good luck to you. Hopefully I'll see you next Saturday. And again, if you have questions, stick around and I'm happy to answer those.